What if aging isn't your body breaking down, but your cells forgetting who they are? Not metaphorically, literally. A liver cell that slowly stops acting like a liver cell, a neuron that starts drifting toward an identity it was never supposed to have. Dr. David Sinclair at Harvard spent 3 decades asking why we age, and the answer he found sounds less like biology and more like a software bug. Here's the mental model you need. Your DNA is the hardware. It barely changes over your lifetime. The actual genetic sequence, mostly intact. That's not where aging happens. What ages is the layer on top, the epigenome. The chemical annotation system that tells each gene, turn on, turn off, stay quiet, go loud. Think of it as the operating system running on top of the hardware. Now here's the problem Sinclair identified. Every time your cells suffer microscopic damage, and they do constantly, your body sends in repair crews. Proteins called sirtuins drop whatever they're doing, rush to the damaged site, fix the break, and then try to go back. But try is doing a lot of work there. After thousands of repair cycles over decades, those proteins don't return to their exact original positions. The epigenetic landscape gets scrambled. Bit by bit, the annotations drift, which is a very clinical way of saying your body's IT department is slowly burning out. And nobody's filing the tickets anymore. And a cell that can't read its own instructions clearly starts to forget what it is. Sinclair calls this the information theory of aging. The hardware is fine. It's the software that's corrupting. So how do you prove that? How do you show that epigenetic chaos causes aging, not just correlates with it? You break it on purpose. In 2023, Sinclair's team engineered mice with a biological switch. When activated, it triggered constant, targeted DNA breaks. Not mutations, just breaks, the kind your body fixes every day. The repair machinery activated, sirtuins mobilized, damage was fixed. But after 3 weeks of this, just forcing the body to keep repairing, something remarkable and disturbing happened. The mice aged. Fast. 3 weeks. The equivalent of years of accumulated biological wear compressed into a single month. Graying fur, muscle loss, cognitive impairment, brain inflammation. Their epigenetic clocks, the molecular timestamps inside their cells, had lurched forward dramatically. And here's the detail that should stop you cold. The DNA itself was fine. No mutations, the sequence was intact. The only thing that had changed was the epigenetic layer, the software. This confirmed what Sinclair had theorized. Aging isn't caused by DNA damage. It's caused by the response to DNA damage eroding the system that keeps cells coherent. But here's where AI enters the story, because without it, we'd have no way to actually verify any of this. The tool the researchers used to confirm the mice had biologically aged? An epigenetic clock, a machine learning model trained on thousands of DNA methylation samples, those chemical tags on the epigenome, to predict biological age from molecular patterns alone. The original version, built by biostatistician Steve Horvath, identified 353 specific sites on the genome whose methylation pattern predicts how old your cells actually are, regardless of your birthday. Newer versions, DeepMage, GrimAge, are deeper neural networks trained on larger datasets. They don't just tell you how old you are, they tell you how fast you're aging and what you're most likely to die from. They also developed their own AI-based physiological clocks, such as Fright, which predicts chronological age, and Afraid, which predicts life expectancy. When Sinclair's mice aged in 3 weeks, the clocks caught it. And when the cells were later restored, the clocks confirmed the reversal. Now here's where it gets genuinely strange. If aging is information loss, scrambled epigenetic instructions, then maybe the original instructions aren't gone. Maybe they're just buried. Sinclair's team tested this with partial cellular reprogramming, using 3 transcription factors called OSK. Delivered via gene therapy into the eyes of damaged and aged mice, they essentially asked the cells to reboot. Not fully. That's the key distinction. Full reprogramming wipes a cell's identity entirely. That causes tumors. What Sinclair's team did was partial, enough to scrub the accumulated noise off the epigenome, not enough to make the cell forget it's an eye cell. The result? Optic nerve regeneration in damaged mice. And when they ran the epigenetic clocks again, the timestamps had rolled backward. The cells didn't just function younger, they measured younger. The backup copy was still there, they just needed to hit restore. This isn't just happening in your liver or your eyes, it's happening in your neurons. And the brain has a particularly nasty version of this problem, because it runs a feedback loop. Here's how it works. As neurons age and their epigenetic instructions get scrambled, they start misreading which genes to activate. Some of the genes that accidentally get switched on are inflammatory signaling genes. The cell starts broadcasting a "I'm damaged, send help" signal, not because there's an actual emergency, but because the instructions are corrupted. The brain's immune cells, microglia and astrocytes, respond to that signal and activate. That activation is neuroinflammation, but here's where the loop closes. Chronic inflammation then stresses the cells further. More stress means more DNA breaks. More breaks means more repair cycles. More repair cycles means more sirtuin displacement. And more displacement means the epigenome gets scrambled faster. Corrupted software triggers a false alarm. The false alarm accelerates the corruption. The ICE mice showed exactly this. Artificially scrambled epigenomes led directly to hyperactivated microglia, brain inflammation, failed memory tests, and lost spatial navigation. The cognitive profile of animals years older than they actually were. And now here's the behavioral piece that should genuinely unsettle you. Stress, chronic sleep deprivation, and social isolation all feed into this loop from the outside. They each trigger low-grade inflammatory signaling, which means they're not just making you feel worse, they're nudging the cycle. They're accelerating the very drift that causes the problem. Loneliness doesn't just feel bad, it shows up in the methylation patterns. It's a molecular timestamp on your genome. Here's where we are in 2026. We can now measure biological aging in real time. Because AI learned to read the epigenetic log your cells have been keeping your entire life. We can accelerate it in mice on purpose to study it. And in controlled conditions, we can reverse it, and the clocks confirm it. The limiting factor isn't the biology anymore. It's delivery mechanisms, dosing windows, the translation from mouse retina to human brain. But the conceptual question, is aging fundamentally reversible, has an answer now. Sinclair's answer, yes, because aging is information loss. And information, unlike matter, can be restored if the backup exists. The question is how long until we figure out how to read it reliably. This is Cortex and Code, where the line between your brain and the machine keeps getting harder to find. Please like and subscribe if you like the video so that more people can learn and grow together. Thanks for watching.